Hello, everyone. Welcome to this edition of the Archaeological Science Interview Series, sponsored by the Society for Archaeological Sciences, also known as SAS. My name is AJ White. I'm a grad student at UC Berkeley studying anthropology, and I'm also an SAS student ambassador, and I'll be hosting today's interview. I'm joined by Dr. Camilla Speller, Associate Professor of Anthropology at the University of British Columbia and President-elect of the SAS. So before I go any further, Dr. Speller, thank you so much for being here today.
No, thank you, AJ. I'm looking forward to chatting with you.
Excellent. So I want to start by getting into your research area, and you describe yourself as a molecular archaeologist. And I'm going to go out on a limb and say that most people, including archaeologists, don't think of molecules as their top thing on a list of words associated with archaeology. So first of all, what does the term molecular archaeology mean? And then secondly, how does it expand the way we think about what archaeology is beyond, trowels and in a bin, traditional methods?
Yeah, thanks. That's a great question. And I'm not actually sure if molecular archaeology has a true definition or not, but I like to think of it essentially as recovering traces of molecules, in my case, biomolecules, so molecules that are created by biological organisms, and studying those in archaeological contexts. So I focus on two biomolecules in particular. First of all, ancient DNA. So looking at genetic material that's preserved in archaeological remains. And then more recently in my career, I've also delved into looking at collagen and other ancient proteins in the archaeological record. So my work essentially tries to recover these biomolecules from archaeological remains, usually archaeological animal bones, but I've delved into other kinds of substrates. And I use those to understand human behavior. So this is why I consider myself first and foremost an archaeologist. And that's because really what I want to do is study human behavior, how humans interacted in the past, especially their relationship to their environment, how they might have changed their physical world around them, how they affect the ecosystems, and then also some of the knock-on consequences of those environmental changes that have occurred all the way up into the present.
Great. And what I love about that description is that archaeology is still there, that archaeological science is answering archaeological questions, but you're using, molecular chemistry to do that. And I'd like to next get into your work that you mentioned on ancient DNA. And this is something that I'm not very familiar with. I'll just put that out there. And I think most folks, including myself, when they think of ancient DNA, we think about its use on humans in the past. But your papers deal mostly with aDNA of animals. So I'm curious if there are any methodological differences in using aDNA on animals than with humans. And then as much detail as you want to get onto this, but how have you used this method to learn about how humans have interacted with animals in the past?
Great, that's a big question. So if I miss a part of it, just remind me and prompt me. Well, I actually, when I first got into ancient DNA analysis, I really wanted to work on human remains, and I especially wanted to work on ancient diseases. I was like, oh, this is what I want to work on, tuberculosis or some other really sexy disease. But when I talked to my supervisor, Dr. Donya Yang at Simon Fraser University, when I was starting my master's degree, he gave me some fantastic advice, which was that working with humans, and especially working on ancient pathogens is it's very difficult. It's subject to a lot of problems with contamination. So contamination from human researchers. It's also susceptible to contamination from the burial environment, which can affect your results for looking at ancient pathogens. And he said, and there's lots of ethical issues, working with ancient human remains. We want to make sure that we're doing that work responsibly and ethically, that we're not destroying human or ancestor remains without being really conscientious about what we're doing. So he gave me fantastic advice, which is start with animals, Camilla, get some experience, learn what you're doing first, and then we'll talk about working on ancient humans. Well, I started with ancient salmon, looking at Pacific salmon at the site of Keatley Creek. And I realized then that I didn't need to look at humans, because actually there's a whole world of super interesting questions about how humans interact with their environments, which we can address by looking at ancient animal DNA. And so I kind of got really lured into the world of animal genetics, and I haven't really looked back. I've done a little bit of dabbling with humans, but more recently in my career, I've really stepped away from that because I recognize that, especially when we work with descendant communities, there is a ton at stake when we interpret the past of people using genetic resources. And so I think to myself, because that requires so much responsibility, I really will leave it in the hands of experts. This is not an area in which we should be dabbling. It should be left to people who are really engaging in that research, who are really making it their research focus, who have the tools and the expertise to do that. And so I feel more comfortable in the animal realm. And so I've stayed there, and there's tons of really exciting questions that we can answer there. And I think the second question was about, what have we, can you repeat it? It's like, what have we learned?
It's kind of the worst question ever presented interview. Like, what have you learned from your body of research? I guess maybe you could throw out some examples of this association of animal aDNA with understanding people in the past and going from that very, hard data about animals to understanding, past cultures.
Yeah, so I'd say there's a kind of two, there's more than two, but in my work, there's kind of two major things that I've learned. One is that I've really used DNA as a tool for species identification. So this has been a key component of my work from the beginning, is using the genetic material that we have preserved in archaeological remains to get really precise and accurate information about the taxonomy of animals, the species, but also the populations that lived in particular areas in the past. And so that allows us to understand at a far more nuanced level how humans are interacting with various species or various populations of species, how the ranges of those animals might have moved associated with human behavior or associated with natural or climatic events. We can look also at how the behavior of humans, like hunting, for example, or overhunting or commercial harvesting, has affected the genetic diversity of animals. And the powerful thing for me about genetics is that we actually have the ability to really match archaeological and modern data by using genetics. By looking at the genomes of animals, we can look at the past genome and at modern genomes, and we're talking to each other at the same scale and using the same language and using the same techniques. And so I think that's a really powerful window we have into the past when we're able to get, kind of compare like for like with genetics at high resolution. And I guess in terms of another way of thinking about how, what insight this has provided about humans has come from my work on domestication. And I find this really fascinating because this is a relationship between humans and, in my case, animals, where humans are really manipulating animal populations. And animal populations sometimes benefit from that manipulation and sometimes they don't. But I remember being completely taken aback when I first started studying turkey domestication in the Southwest United States. That was what my PhD was focused on. And when I first started analyzing these turkey bones, we discovered that I was studying hundreds of turkey bones from the Southwest United States, and they all had the same, almost all of them had the same mitochondrial signature. And what this was showing me is that humans really controlled the breeding of this animal. Domestic turkeys were introduced into the Southwest, and then from then on, they were sort of really maintained and bred, and you could really see the human control over these populations. And that was one of those first inklings I had, wow, this is such a fantastic technique that we can actually see how humans are confining and penning their animals and restricting their breeding, at the genetic level. And then since then, genomics has really exploded, and now we have the potential to look at in a really fine-grained way how humans manipulate animals, especially domestic animals. So it's an exciting time to be an archaeological scientist working in genetics.
Yeah, absolutely. I realize now, I'm like, gosh, that is a long list where we could spend the whole rest of the interview talking about each one of those subject areas. But for the purposes of our talk today, I'm going to move on to a question about your career trajectory And you mentioned how you've worked on a number of animals, starting with salmon, and I've got a little list prepared. I think I missed some, but beyond salmon, we've got turkey, water buffalo, horses, herring, cattle, elk, dogs, whales, mammoth, a little bit of humans you mentioned as well. And if I missed some, you're welcome to fill in the blanks in just a second.
There's some sheep in there. There's some
okay, sheep.
calculus. There's lots of things. Yeah.
So with that long list established, I think that most students, in archaeology, particularly archaeology science, want to do that. Want to take their method that they learned in graduate school and apply it and just totally expand our knowledge base, working in not just one type of information, but just multiple places throughout the world, multiple times. So I'm curious if you have any advice for emerging, archaeological scientists on how to situate their careers for that ability to really proliferate and do what I think, we've talked about here.
That's funny, AJ, that you should say this. I'm not sure that I'm a good example. The reason why I have this laundry list of species and also techniques. I mean, I don't just work on ancient DNA. I also do Zooms, which is a collagen-based species identification approach, and then also ancient proteins. I would say that this is not a good idea. So at first, when I started my career, I was looking at mitochondrial DNA. And in those early days of archaeological science, when we were looking mostly at mitochondrial DNA, it was a technique that you could take to very many animal species, because we were essentially using a tool looking at one small, simple genome, the mitochondrial genome. The techniques that we were using could readily be applied to a number of different species. And we could investigate a lot of questions. But since the advent of genomics, I've come to realize now that it's not a great idea to start moving from organism to organism to organism, because the questions that we ask of that genomic material will be different in every case. And it's not as transferable a skill as mitochondrial DNA. The other thing that I grew to realize when I just started saying yes to every project was that by applying this tool in a lot of different places, I could answer a lot of questions. It was super exciting for me as a researcher. I got to engage in a ton of collaborations. I learned a lot. But at some point, I began to feel really a bit uncomfortable that my level of knowledge about each new system, each new culture, each new geographic region that I moved into was more and more limited. And it made me question my ability to really answer questions robustly, to make sure I could authenticate that data and that I was really using the right tools to answer the question. And I realized I was actually in danger of becoming so distant from the archaeological questions and the archaeological context that I could be misinterpreting the results. Now, I always work closely. I collaborate with a lot of archaeologists, and those colleagues are wonderful for making sure that the technique is grounded and the results are robust. But I did start to feel this nervousness that by being a jack of all trades, you suddenly become a master of none. So more recently, since moving to the University of British Columbia, I've really taken a step back and asked myself, what is really most important to me as a researcher? What are the questions that I really feel passionate about? And where do I want to direct that attention so I can do careful and rigorous work? And for me, that is really now thinking about working in marine ecosystems. So I'm really, I started with salmon, I'm kind of coming back to it now. But really thinking about the humans' impacts on marine ecosystems in many different ways. And especially living here on the Northwest Coast, I have a privilege of being able to work with local communities and local Indigenous communities who have welcomed me to participate in the research with them, to help build those questions and that research together, and to be able to make a meaningful, I think, contribution to their lives. There's so many ways in which colonialism has alienated people from their traditional territory, from their ability to procure resources. And if in some small way I can use these techniques to try to rectify some of those injustices, then I really, it's a privilege to be able to do that. So I guess if I had some advice for up and coming researchers, is that it is great. It's a wonderful opportunity to be able to take your skill set and apply those in different areas, but to really be conscious about not saying yes to everything, not every opportunity, but to choose one or two areas that you really feel passionate about and that you really want to dedicate your scholarship towards, and so that you can really make a meaningful contribution in those areas.
Gotcha. No, that's much appreciated, at least on a personal level, about being, hey, you gotta do what you do well, right? And then to make those meaningful connections to modern communities and to understand the archaeological record, it does take time, I guess, to do that. So thank you for imparting that advice. Since this is a SAS sponsored interview, I'd like to move to a question now about your role in the SAS now and your upcoming position. Maybe you could state what that is in just a moment, but also could you share how you became involved with SAS?
Yeah, so I feel a little, I feel new to the SAS, even though I joined many years now. And I joined principally because they had this wonderful deal at the SAS one year, which was that if you joined the SAS, you could publish in, I think it was STAR, the Science and Technology and Archaeological Research Journal, for like the open access fee was $25 if you joined the SAS. So it was amazing, or something like really, anyways, this is really sweet deal. And so I was like, this is brilliant. And I've always enjoyed reading the SAS bulletin. And so that's why I kind of kept up over the years with the SAS, but really as kind of from a distance. I hadn't really appreciated all the things that the SAS does for graduate students and early career researchers, advertising positions and offering awards and, co-sponsoring conferences and so on. And so really my involvement with SAS really expanded only this year when I was elected president-elect. And that was almost by accident. I was invited by Vangelio and Marcos to consider throwing my name in the ring. And I thought, oh, wow, this is such a great opportunity to be able to reach more early career researchers, to be able to support archaeological science. And so it was really kind of unexpected. It wasn't a career plan or anything like that. I just thought, oh, what a wonderful opportunity. And, I'm going to go for it. So I'm still learning a lot right now about the society, and I'm loving everything that I'm learning. And my colleagues are amazingly dedicated. So it's a really wonderful team to work with and learn from. So I'm still in the very early stages of learning so much more about the society, but I'm excited to keep that going.
Well, thank you for sharing about your trajectory there, and congratulations on your position. We just have one more question today on our list here, and it's admittedly a cheesy one, but what have you enjoyed most about working as an archaeological scientist?
Yeah, I know. It's kind of funny because you asked me in advance to think about this, and I thought, man, it's so hard to pick just one thing. And I guess reflecting on it, I'm sure many people probably have the same answer, which is that, when I was a student, I loved being a student because I loved learning. Like I feel curious about the world, and I love, finding things out. And so being an archaeological scientist is like this privilege that allows you to keep being a student your whole life, in the sense that you're always investigating something new. There's always a new puzzle to be solved. There's all these questions out there where you can apply your technique and maybe reveal some new piece of a puzzle. And so that's what I think I love about archaeological science. Plus I love history and archaeology and human behavior. And so it brings it all together, but in a way that I like to not trick myself, but I like to think that we bring a little bit of objectivity, to this question. And admittedly, although we all come with our biases and, we can never be totally finding an objective truth. I like the fact that there is this, scientific technique that we can apply to these different questions, at least to investigate human behavior in a new way. So for me, it's all about the curiosity and the puzzles that keep me going in this area.
Well, I'm excited to hear about the next puzzles that you tackle. in your career, and just want to say thanks once again for sharing your time today to tell us about your story as an archaeological scientist. For everyone else, please subscribe to the SAS YouTube page, and thanks very much for watching.